All right, I might start introducing you, Marissa, and we might get a few latecomers, but yeah, okay, great. Well, welcome to another Friday seminar. Really pleased to introduce Dr. Marissa Betts from the University of New England. She's a postdoc and a lecturer there in the Paleoscience Research Centre. She's a paleontologist. She specialises in the Cambrian, especially investigating links between evolution, ecology, and timescales of those changes. She's done lots of really cool field work in South Australia, but also in Canada, Mongolia, China, and the US. And she's also a passionate science communicator and known for doing various sort of radio interviews and podcasts on Paleocast. Is that right?
Yeah, that's it.
Yeah, Paleocast and various sort of SciComm posts. And she's also a STEM coach for the Curious Minds, which is aimed at trying to get more girls into science. So that's really, really great. So yeah, the title of our talk today is Tackling Big Cambrian Questions with Small Shelly Fossils. So take it away whenever you're ready. Thank you.
Oh, thanks, Lucy. And thanks very much for inviting me to come and give a Zoominar in your seminar series. I've never given a Zoominar before, so I hope this goes well. So I'll launch into it. So I often like to start with some of my talks, especially when I'm dealing with shelly fossils, with this particular slide. And it shows lots of different small shelly fossils, Cambrian small shelly fossils that I've gotten out of samples from the Flinders Ranges. So these are all Australian small shellies. And if you notice, I hope everybody can see on the bottom of the slide is a scale bar, and that's 1 full centimetre across the bottom. And so they're all to scale, and so you can see that they are very small. They live up to their name. And you can also see that there are some forms there that you might recognise. So things like this one at the back is a brachiopod, member of a group that is alive today. The mollusks as well here. And this one also a mollusk. You can see very clearly this has a very snail-like shape. But then there are a whole host of others here that you might not so easily recognise, and they can be kind of classed as more problematic taxa. And a lot of the history of research into the small shellies has been kind of about what they are or what they were. And so putting them together like a puzzle has been a very big area of research for a long time. So you can kind of tackle small shellies from 2 main angles, which is what I wanted to talk about across both of these sorts of things in this talk. And one is kind of a paleobiological angle, so looking at what they were, their phylogenetic relationships, how they built their skeletons, and then also the sort of applied side, how we can use them to answer more broader questions. And they're a really important part of the Cambrian explosion, and I guess that this sort of thing doesn't need terribly much explanation for this crowd maybe, but it is a really important period interval in the history of life on Earth, and it's where we see the very abrupt appearance of lots of different kinds of animal body plans kind of all at once in the fossil record. So lots of all of the major phyla, the major groups of animals that we know today, have their origins all the way down in the Cambrian explosion in the early Cambrian, 542 or so million years ago. And one of the really important aspects of the Cambrian explosion was the development of biomineralization. So this is where the shelly fossils sort of come in, the skeletal animals. And there's lots of really good reasons why developing shells or biomineralizing shells is a really good idea. But one of those is that it allows organisms to develop stronger, larger, and more complex bodies. Another good reason to have a shell or an exoskeleton is for protection or defense against predation. And so in the Cambrian, we do see the development of these sort of complex ecological interactions like predation, and the shelly fossils are a really important part of that story as well. So really the fundamentals of all modern ecosystems, everything we know about life in the modern day really has its roots all the way back down in the early Cambrian. And I've seen the Cambrian explosion described in all sorts of different ways, and people sometimes try and be as evocative as they can. And my favourite way that the Cambrian explosion has been described was in a textbook, and they described it as an astonishing orgy of evolution. And it always gives me a giggle. I really love the words astonishing and orgy right next to each other. So I also wanted to talk a little bit about preservation, just to sort of orient ourselves in the sort of small shelly world. And it's important to think about how they're preserved. The shellies fall into sort of two camps. The originally phosphatic ones, so things that biomineralize calcium phosphate, and the ones that are secondarily phosphatized, so things that have been sort of infilled with phosphate or phosphatic mud or coated in phosphate. They're recovered almost exclusively from carbonates. We don't really see shelly fossils coming out of siliciclastics, especially coarser things like sandstones. The reasons for that are probably twofold. It could be that those kinds of paleo environments weren't necessarily so great for the shelly fossils, and it's probably also a taphonomic sort of thing as well. When we do see the shelly fossils in the carbonates, they're definitely elevated. Their concentration is definitely elevated in these things. We see hard grounds and firm grounds, where we see a lot of phosphate being deposited. So these are kind of hand-sized samples from the flinders, and you can see in these polished sections these grey sort of horizons, and they're omission surfaces where you don't get so, or sedimentation slows down and phosphate starts to sort of build up there. And you can see it really nicely in these elemental maps. So these are done with a Bruker tornado at Macquarie, and the phosphate's brought up in this beautiful green, bluey green colour. So you can see very well these surfaces and how they've developed sequentially over time, and also how they've entrained the shelly fossils. These are the shelly fossils that come up really, really nicely, and you can see they've been infilled with phosphate. This is work that I sort of did with my my friend and colleague Sarah Jacquet, who is now at the University of Missouri. And she put this stuff in the SEM, these polished sections, and showed that these fossils here, this is a bivalve mollusk and this is a univalve mollusk, and shows very clearly that they've been infilled with phosphate. The shells are actually still calcium carbonate. This is calcium here. But when we get them out of our residues, we only get the internal mould, and that's what this looks like. And so it's important to think about these processes because there are all sorts of biases involved with their preservation. So, you know, if you think about taxonomically, these things have been identified to species based on the morphology of what is essentially the inside of the shell. And then also for applied things like biostratigraphy, how effective are they as biostratigraphic tools if we only really see them being preserved in association with these phosphatic facies? So these are the sorts of things that we must sort of stay in touch with throughout the whole process of investigating shelly fossils and applying them for to bigger questions. And she's also done some really cool stuff with CT. So Sarah's work is really looking at shelly fossil taphonomy and preservation. And so she has access to a cool micro CT, and you can see this sample I actually brought back from Mongolia and gave to her. And the phosphatic fauna are very beautifully preserved throughout there, like raisins in a cake. So once you do get these processes happening, then the shelly fossils are very abundant. So the things that are originally phosphatic, the things that biomineralize calcium phosphate, are usually the things we call multi-element animals, in that they had a skeleton that was built of lots of different little bits and pieces. And we call those little bits and pieces a sclerite. And in our assemblages, they're usually found as separate elements. But in life, they would have articulated together to make a suit of armour which we call the sclerotome. So the sclerotome is made up of lots of different little sclerites. I throw this in to sort of talk about our fieldwork and methodology in finding and extracting the shelly fossils. When I tell people that I'm a paleontologist, and most paleos probably have this, that the first thing that people think about is Jurassic Park. And people assume straight away, of course, that I work on dinosaurs, and I have to disappoint them greatly and tell them that I don't work on dinosaurs, unfortunately. And when I talk about field work and doing paleontological field work, this is the very first thing that they think about. The sort of scene in Jurassic Park where they're sitting around brushing the dirt very gently away from the velociraptor skeleton. And of course, because we work on very small things, our field work looks very different, and it looks something like this. These are actual field work photos. This is the person on the bottom. It was my PhD supervisor. This is Professor Glenn Brock at Macquarie University. He's in the Flinders here in the bottom right, and in Antarctica on the bottom left. That's in 2011, and I'll talk about the material they collected during that trip. I'm working on that with them. So we do have to get in very close to have a look at the rocks, because even though a lot of them are microscopic, some of them are big enough to see with the naked eye. And one of the sort of more obvious fossils that we would see in the carbonates that we're looking at are things like Archaeocyaths. So these are a sponge-like organism that built the very first, well, animal-built reefs. And they were really important ecosystem engineers. So they created these new sort of ecological niches that the shelly fauna started to sort of expand into. The thing on the top left here is Micrina. This is an originally phosphatic shell, and you can see that it's popping out of the limestone. So the limestone's weathering away, and we get the shell kind of popping out, and it's quite large. And then on the right here, we have a steinkern from mollusk. This is the material from Mongolia that that Sarah put in the CT before. And so all of our material really is measured taken from measured stratigraphic sections. So this is the Bunkers Range in the Flinders Ranges. A lot of you probably know this area very well. And you can see that the Cambrian here is exposed beautifully. For kilometres and kilometres, you could follow these beds. And so we take measured stratigraphic sections for as long as we can, up to several kilometres long, and we sample at regular intervals along that section. And the material is brought back to the lab, and then it is acid etched. So we put them in weak acetic acid, so it's just a vinegar, a little bit stronger than table vinegar, and the material is etched away, and the insoluble stuff is sieved and dried, and then we pick it for the microfossils just with a tiny paintbrush. But it's not until you get the material under high-powered microscopy that you really get a good sense of the assemblage that you have. The SEM is really important for identifying some of this stuff to species level, because they have sometimes very fine micro ornament on the outside that is important for identifying different species. And this is really where the work starts to get exciting, because you can work out what you have and what you can do with it. So from this point in the talk, I'm going to talk a little bit about one particular microfossil, a small shelly fossil, and its paleobiology, work that I've been doing in that sort of area, and then I'll talk a little bit more about applications. So we're going to zoom into this particular thing in the top right here, which is Microdictyon. I've been working on Microdictyon with a colleague, Michael Steiner, in Berlin. He's at the Free University of Berlin, and I was funded to go there for a couple of weeks last year to just look at this particular fossil in great detail. I've got the Australian material. He's got Chinese stuff and things from Kazakhstan as well. And this fossil, Microdictyon, has been coming out of the Cambrian acid residues all over the world for a long time, and people had absolutely no idea what kind of creature could possibly have produced these sclerites. They're similar but not always the same. They're kind of round or quadrate, and they were described originally as being perforate. When you get in close with the SEM, they have these beautiful nodes around these holes. And the sort of morphology of these different nodes is how people have been designating different species. When you flip it over, you start to realise that actually they're not holes, it's not perforate, it has these little caps that cover the holes at the back. This thing here is not strictly Microdictyon, but it's very similar, and we have Microdictyon sclerites preserved in a similar way. Now, can you see how the sclerites are sitting one inside the other? And this is a clue that tells us that it's a molting animal, that this thing was caught in the act of molting. So there were lots of ideas about what it could have been, but until they found the body fossil in Chengjiang, this was when they finally realised what Microdictyon was. And it was very excitingly a soft-bodied worm with legs. So this is from the Chengjiang in the south of China. So I was lucky enough to be able to collect there a little while ago. And it's reconstructed something like this. So it is an onychophoran, and it's closely related to modern velvet worms with their lovely little soft legs. And you can see where the sclerites are. They're in pairs down the body, and each pair is sort of associated with the top of a pair of legs, except it has 10 soft legs but only 9 pairs of sclerites. So the Microdictyon's kind of one of those examples with Cambrian shelly fossils where people talk about it as a success story, small shelly success story, where we had the sclerites and we didn't know what they were, and then once we found the body fossil, we knew that it came from a soft-legged worm like creature, and that's it. The problem is solved and everybody can go home happy. But there are lots of things that we still don't know about Microdictyon. Maybe if I just go back, the very tiny details that you can see with the SEM on the sclerites are not preserved in the Chengjiang material. Those are the ways that we identify species. So that we have no way really of connecting the material that we get from Chengjiang with the material that we get out of the acid residues. We don't know if we have this particular species from China in the acid residues in Australia or Kazakhstan or anywhere else. I also had a look very closely with Michael in Berlin at the way this thing was preserved. And when we get close up, you can see that it has this kind of a capping layer on the outside, which in the mapping, elemental mapping, is very rich in phosphate. So it's this almost like enamel on your teeth. And underneath is kind of spongy. And we etched off this phosphatic capping layer and revealed these sort of these structures underneath. We kind of started to call them brushes. This is in cross section here. And as far as I know, I've never seen that before. I don't think it's ever been documented. And I couldn't tell you really what these things are for. This is in the Australian material and also in the Chinese material. So it seems to be happening in all of the different specimens that we had a look at. So we don't really know what these structures were for. Nobody really knows what the sclerites were for. One of the more creative ideas is that they might have been eyes that occurred in pairs all the way down the body, but developmentally that's a little bit difficult to reconcile. No one really knows how it grew, so we have tiny specimens and big specimens, and in the middle, not really sure what happens. People don't actually really know which end is the head, which seems like a fundamental thing to be able to say about a fossil that you're studying. And at the end of the day, people don't really know how many different species there are. And this is sort of the important point for the applied work. So it's really important to be able to identify species properly, to be able to apply them to any sort of bigger or a bigger question, right? So in terms of working out whether the same species was globally distributed with microdictyon, we just don't know. And so therefore it's not going to help us terribly much for answering bigger sort of questions. So I just wanted to talk about this sort of paleobiological investigations and how important it is to really to spend time to answer these very tiny questions, because they have knock-on effects for bigger things. Okay, so the rest of the talk is more about my applied work and chronostratigraphic sort of work. So everyone knows this diagram, the geological timescale. If we go down to the Cambrian, we've got some problems. The Cambrian is the only period in the timescale that still has unratified series and stages. So there's some really important boundaries in the early Cambrian that haven't been ratified. We've had recent success. Last year they ratified the middle Cambrian boundary, so now it's the Meolingian or the Woolioan boundary. But the main burst of the Cambrian explosion really happened down here. And if you want to answer any questions about the timing of important evolutionary events or the tempo of the Cambrian explosion or any of those things, you need to have a working timescale. And at the moment, we still haven't quite got there. In Australia, the situation has been quite bad for a long time, in that we don't actually have any stage subdivisions at all. So this is Australia here, and the different subdivisions in the different regions around the world. And this sort of lack of subdivisions has really isolated the Cambrian successions in South Australia from being integrated really into a global chronostratigraphic or temporal synthesis. So we've never really known exactly where we sit in relation to everywhere else. So we kind of tackled that problem using a multi-proxy sort of approach, which has been advocated by the International Commission on Stratigraphy. Biostratigraphy is really the classic way that people sort of build these chunks of time into the timescale. And now chemostratigraphy is almost as widely used. And of course, if you can get high resolution or highly accurate radiometric dates, that's very important as well. So this is the sort of South Australian work and building the sort of new scheme that we've been using. And the work is based on, well, I did most of this work for a large part of my PhD, but the material that I used was part of a huge database that had been sort of systematically worked on and collected by Glenn Brock at Macquarie and his sort of colleagues and students and whatnot for, I guess it would be 10 to 15 years of concerted effort collecting in South Australia. And I mention that because it's very important to say that the work is built on very recent taxonomic assessment of the Shelly fauna from South Australia. So I said before, it's really important that we understand the species that we have to be able to use them for any kind of applied purpose. And that work has been done very recently, so that's very important. The biostratigraphic data is from 25 measured sections and drill cores in the Stansbury Basin and also the Arawi Basin. And then we subsampled select sections to do the chemostratigraphic work. And that was done in the University of Adelaide. Tony Hall did that for us. So that was really great. And we also have ID TIMS dates from both basins that were done by Jim Crowley at Boise. So these are the sections from the Stansbury Basin here as well. So this, the next couple of slides just sort of summarises the new scheme that we built with the Shelly fauna. We noticed that we used what we could clearly see was a repeatable pattern with the stratigraphic occurrence of the Shelly fauna. And we described or erected these new zones in two publications in Gondwana Research. And then we back that up with integrating the biostratigraphic work with the chemostrat and the radiometric dates. So I like to think of it as a trilogy. So all of it is taken together. So the oldest zone that we were able to identify is this assemblage of taxa. So this is the Culparina rostrata zone. Culparina is this ugly beast at the top here, very crusty, a multi-element thing. And it's accompanied by a very low diversity assemblage, but we have brachiopods and other shelly fossils as well associated with Culparina. The second zone is the Micrina etheridgii zone. That's this one here. In this zone, we have a more diverse assemblage. So we have the incoming of mollusks here, arthropods, and the very first trilobites, or the oldest trilobites in Australia in this zone. And the third zone is the Daleatia odysseyi zone, and that's this beast here at the top, the first four there. And this is a very high diversity zone, and with some species actually that are globally distributed. So we have a good chance to globally correlate this zone. In the Cambrian, a lot of the faunas are very endemic around the world. So when we have globally distributed species, that can be really, really critical for correlation. So that's the succession in the Arawi Basin. This is we can see the same three zones in the Stansbury Basin as well. But I just wanted to talk a little about the lower parts of this succession. If I just go back to the Arawi Basin, you can see the very oldest units here are the Uratana and the Parachilna. And these are siliciclastic. But in the Stansbury Basin at the same sort of level, the same time, these units are carbonate, or they're carbonate rich at the very least. And so that means, like I said before, how we only really get the shelly fauna from the carbonates. It means that we get a different, older shelly fossil assemblage in the Stansbury Basin that we just don't see in the Arawi Basin. So in the Stansbury Basin at this level, we have Watsonella crosbyi, that's this guy, and Aldanella. And the really cool thing about these two is that these are globally distributed taxa. So unlike most things in the Cambrian, you find Aldanella and Watsonella in almost every Cambrian terrain at around this time. So they're really, really important for global correlation. But most of our fauna are endemic, and so the really critical thing is to work out a way to really integrate our successions into a global scheme. And the chemostratigraphic curve, or the carbon isotope chemostratigraphic curve, is it more readily enables global correlation, but you need to use it sort of with a grain of salt, I guess. The isotope curve, or the Cambrian carbon isotope curve, is characterized by these very large amplitude sort of excursions. But actually working out in your sections which peak you have can be quite difficult, because you can see that there's quite a bit of variation in the magnitude of these events. And so the biostratigraphic data is really important to help you kind of identify which peak you are looking at. So we've always sort of said that using chemostratigraphy and biostratigraphy together is better. So in our sections, this is sort of an example of how we kind of went about it. The really reassuring thing that we saw was that there was a repeatable relationship that we could see between the biostratigraphic zones and the chemostrat. So, for example, this is two sections in the Arawi Basin. At this point here, the boundary between the Micrina zone and the Daleatia odysseyi zone, the isotopes tended to have this positive sort of a trend, with a peak here that was always above this boundary. And we saw that pattern again and again and again. And so we were able to correlate this event, this positive event, with the CARE, which is the Cambrian arthropod radiation event. That's a global event. And we could definitely start to see the same sort of trends, the global trends in our data as well. In the Arawi Basin, like I said, lower than this, we didn't have any carbonate, so we couldn't get any results. But because we do have carbonate at those levels in the Stansbury Basin, we predicted that we would see isotope results that look something like this. And we were very relieved to see that we did in fact get similar sorts of results. But any lower than that, in the South Australian successions, we don't really have any carbonate, and we also have a massive unconformity. We don't really get Fortunian rocks in South Australia, I don't think. And so that's probably as low as we can go. So putting all that together, we're able to do a reasonably good job with integrating our successions now kind of with, or correlating them with the other regions and putting them into a more of a stronger global context. And a really important part of that was the radiometric dates. So we have these ID-TIMS dates, and they all kind of clustered around this kind of time. So 514, 515, and up to 511, 512. There are more tuffs in the Billy Creek that haven't been dated. That particular one is in the lower part, but there's some in the younger or the upper part of the Billy Creek that we're going to get dated, which will be an important part of that story to take a little bit higher. One of the kind of controversial outcomes, I guess, for this work was this controversial placement of the Parabatiella Huai zone. So this is the oldest trilobite in Australia, and actually the oldest trilobite in East Gondwana. It's the same species as we get in China. There was sort of this prevailing wisdom, I guess, when I started my PhD, that the trilobites, the oldest trilobites in Australia, appeared much later. They were very diachronous. They appeared much later than elsewhere in the world. And trilobites are sort of meant to come in everywhere at around the stage 2-3 boundary. So here, and that's the trilobite zones here. But our work has suggested actually that the incoming of trilobites in Australia is more in line with where they first appear elsewhere in the world. And we have a new sections as well to try and kind of capture this again, and the results are saying something very similar. So I didn't set out to be controversial, but this is sort of what the data is saying, and I think people might just have to get used to it. So there are some interesting paleobiogeographic signals that the Shelly fossils are giving us. So we're using them for the correlation as well, but it's the similarities between some of these terrains can maybe tell us some things about paleobiogeography. And the fauna from South Australia is very similar, obviously, to Antarctica. We were very close at the time. Laurentia, South China, and also North China. Baltica and Avalonia down here, maybe not so much. It's a different latitude, so up here is sort of more tropical. It's colder, maybe latitudes here. Baltica actually as well is very siliciclastic at this time, so you don't really get very many shelly faunas anyway. But there's some really interesting stuff that has come out of North China recently. And so I was saying before that the South Australian shelly faunas, a lot of them are endemic, and we don't see them anywhere else, except maybe for Antarctica. So imagine our surprise when some of the things that we had only ever seen in South Australia started to turn up in North China, and these are the exact same species. So all of these are the exact same species, and they occur in South Australia and North China. And so I think that has something important to say about the sort of reconstruction of the Cambrian world, especially for the position of North China, which has been apparently a mystery. So these are 6 different reconstructions here that show 6 different positions of North China. I did watch the talk last week, and some of these reconstructions were described as motorboat tectonics. which I thought was really funny. I could imagine these sort of terrains sort of putting around the globe. I think this is actually the only real tectonic reconstruction here. The other ones are probably just sort of eyeballed. But this here, I think, is done mostly with paleomag, and it whacks North China way out here. And the shelly fauna would suggest that this might not be right. And so they could have something really important to say about sort of constraining maybe, or these kinds of reconstructions. So they could have a very important role for paleobiogeographic or even tectonic reconstructions. Okay, so I guess paleobiogeography is a segue here. The next kind of few stories are about correlation work and chronostratigraphic work in Antarctica, and also my work in Mongolia. Glenn and his colleagues went to Antarctica in 2011 and collected long measured stratigraphic sections there. Australia and Antarctica were part of Gondwana during the Cambrian, and around that time they were sort of up around the equator, so the climate was obviously very different to today. Cambrian rocks in Antarctica have been known about for a long time. This is one of the oldest papers I've seen from 1921, which documented these things, the archaeocyaths that had been dredged from the Weddell Sea during the Scottish National Antarctic Expedition. So this was at around the same time that Scott was heading for the pole. So they'd known about the Cambrian there for a long time. A lot of information about the shelly fauna has come from erratics, these glacial erratics in a Miocene formation on King George Island, so that's out there. But you know, these things have given us lots of information about the sort of Cambrian shelly fossil diversity, but because they're just from spot localities, they can't tell us much about, or can't give us much information to help us correlate these rocks terribly well. So this is Glenn, and also Lars Holmer and Paul Myro from Colorado, Christian Skovstedt from the Swedish National History Museum, and Lars Stemrich from Copenhagen. And they, what they did, which was different, is that they collected material along measured sections, and in probably was, you know, pretty rough conditions. I would, you know, give me a beautiful winter's day in the Flinders Ranges any day. But they measured through the Shackleton limestone, the Holyoke Formation, and the Starshot Formation. So this is the bird group in the Transantarctic Mountains. And so I'll share with you some of the results that I've been looking at for them. So I guess based on the success of the South Australian work and my familiarity, I guess, with the successions and the fauna that are very similar to the material in the Transantarctic, so I've been kind of involved in interpreting some of this stuff. So this is about to be submitted. It's very close now. This is the combination of the isotope data and also the fauna. The coolest thing is that we have Dahliatia odicei in the Shackleton limestone. So this is the eponym for the youngest zone that we erected in South Australia. So it's a direct species level connection, and it's also sort of time significant. So it's a really important fossil to find in Antarctica. The fauna from especially the Holyoke Formation and the Upper Shackleton limestone is beautiful, it's diverse. The fauna is very similar to South Australia, as you would expect, but also North China, and also Laurentia as well. There's this beautiful bradorid spine. It's very distinctive, and so we know for sure that we get this in South Australia too. These are trilobites from the Shackleton line, or the Starshot Formation above the Holyoke Formation here, and they're almost identical to a fauna that comes from the Simbrick Vale Formation in Western New South Wales. So this is Starshot here. So this is I guess the first time that we've been able to put the Antarctic Cambrian successions into some kind of chronostratigraphic context like this. And it's been based on the shelly fauna that's coming out of the Shackleton limestone and Holyoke and the Starshot trilobites, and also using the isotopes from the Shackleton as well. And so it's telling us that the Shackleton limestone's probably more or less time equivalent to things like the Mern-Myrna Formation in the Flinders, and the Heatherdale Shale probably or the upper Heatherdale Shale, at least in the Stansbury. The Simbrick Vale has a tuff in the bottom of it. That has been redated. There was a shrimp date that I think came out at around 517, but it's been redated by Jim Crowley. It's not published, but it's come out at something around 515, which is more in line with what you see in the Arawi and Stansbury basins, which is quite nice. So this is the sort of upshot here. This is the correlation for East Antarctica. Oh, and one very cool thing came out of this is that last year I described a new species of shelly fossil from the White Point conglomerate that's classed in the White Point conglomerate. And I called it Daleatia decobruta, and decobruta means handsome beast. And one of my colleagues is working on the transantarctic specimens, and he discovered it in the transantarctic stuff. So it's very cool to see handsome beast turn up in the Antarctic material. Another species level connection. So the last story is about the work that I'm doing in Mongolia at the moment. This is a photo I took in 2017 in Solani Gol. So this is in the west of Mongolia in the Gobi Altai area. And we've done several seasons there now. We were scheduled again to go there again this year, but that has been canned for obvious reasons. So hopefully we can go again next year. We're looking at lots of different parts of the sort of succession in Mongolia. This particular work is looking at the Precambrian, Cambrian boundary and that sort of transition. So a little bit older than what I've been talking about so far. Usually, well, often at this particular level, we have a lot of time missing. There's an unconformity that's often called the Great Unconformity, because there can be over 1 billion years missing at the Precambrian, Cambrian boundary. So identifying the boundary is difficult. I guess even if you have the Great Unconformity, you could just say that it's in there somewhere and, you know, kind of go with that. But if you have a more transitional sort of a section or a continuous section, how do you identify where the Precambrian, Cambrian boundary is? It's a really important, one of probably the most important boundaries in the timescale. So the official way to do it is with the first appearance of the trace fossil Trepticus pedum. And even though it's sort of been said that pedum occurs in lots of different facies, trace fossils can often be, facies controlled, and you don't often get them everywhere. And so there's often a problem with relying on pedum to designate the boundary. In Mongolia, the Precambrian, Cambrian transition may be one of the world's most complete. And so it's a really important place for us to really have a look at what's happening at this particular time, and if we can do something better potentially with shelly fossils or isotopes. So these are the successions that we've been looking at, or the areas we've been looking at. So this is the Gobi Altai area. Last year we went up to the Hoopscore region. It was very wet there, and lots of forest that covers up the rocks. So we're going back to the west next year, hopefully. So this is Byangol, and this is the sort of succession that we had been measuring. Sorry, it's in shadow. We should have taken that photo at a better time of day. There's our little bus down there. There's someone up here as well for scale, maybe. So this is the succession, or the section that we drew up. This is actually just only the lower part of an enormous stratigraphic section that we measured. That's probably close to 2 kilometres long. We sort of started down here, went up and around the corner. So this area as well is very, very faulted. The tectonics of Mongolia is enormously complicated, and on the ground you're always sort of being, you know, there's always a lot of faults that you have to sort of deal with. Bayangol's not too bad, but when you go down here this way, there's a lot of big faults, and we try to avoid trying to correlate too much across faults. It can be quite confusing and difficult. So we stuck to this massive fault block on this side here. The lower part here, this chunky stuff, is the Zunat. So that's blue on the stratigraphic column, and then we have the Bayangol formation sitting on top. The boundary, the pre-Cambrian, Cambrian boundary has often just sort of been whacked in between the two units. So we made sure to sort of target this particular interval. So what would we kind of expect to see across this boundary? This is a kind of an example of what would happen. Sometimes you do see like Ediacaran, but if you get the right facies, those things might creep over. In terms of the isotopes, we would be expecting negative results that would crawl back to a positive. And in terms of sort of metazoans, things like the cloudinids would crawl over that boundary. So we would see something like that, hopefully. So this is the result. So this is in review currently at the moment with geology. This is the first time that the successions from Mongolia, or this sort of age successions in Mongolia, have been sampled synchronously for isotopes and fossils. So even in the last couple of years, it's been mapped very well. There's been sequence stratigraphic work done, carbon isotopes done, but nobody has sampled for fossils in any kind of rigorous way. So this is very new and potentially quite important data. The isotopes do show us that signal from the very negative results that go up to a positive. And we suggest that maybe we have the base excursion. So they always have crazy acronyms. So this is the basal Cambrian excursion, or Cambrian carbon isotope excursion. And that would be the nadir of it there. We're probably not capturing the entire excursion. And with the fossils as well, we have the cloudinids popping in just at the top of the Zunats and then up into the Bayangol formation, so well into what we would consider to be Cambrian time. And these are the cloudinids here, Zunia, recently described actually from the Bayangol formation. Down at this level as well, we also have protoconodonts. So these things, they're very common in lower Cambrian rocks. And then it's not really until we get a bit higher into the section where we get more diverse fauna. Anaborides, also another classic Cambrian shelly fossil, and Cyphogoniacides, this thing, a multi-element beast. Don't ask me what it looks like, I don't think anybody knows. And so we also have the ichnofossils, some ichnofossils, but no pedum really. So we have some sort of horizontal traces. And also this cool thing, Spadangopsis mongolica, sort of star-shaped trace, but no pedum. At this level anyway, there is Tripticus pedum well higher in this section, but to put the Cambrian boundary there would make no sense whatsoever. So I guess there are two kind of upshots for this paper, and one is that in the sections where you get more complete deposition, you start to see this transitional fauna between the Precambrian and the Cambrian that's consisting pretty much entirely of the cloudinids, these sort of annulated tubes. There is an older idea, and I think that it's fading away, that the Precambrian and the Cambrian were these non-overlapping worlds, but actually we do see some sort of transitional fauna that cross over. And then the other question is how to define the base of the Cambrian without pedum. And in this case, we suggested that we would use the isotope event, but in combination with things like protohertzina. So that's how we suggest that maybe it could be done in the more transitional sections without venom. So to finish, I hope people enjoyed that talk, and I wanted to sort of talk about the two sides of looking at small shelly fossils. One of the paleobiological side, where you get to look at phylogenetic relationships and functional morphology and ecosystem structure, and then of course the applied side, biostrat, timescale, development, those sorts of things, with the knock-on effects for questions about paleobiogeography. And also they can teach us things about biomineralization in early animals, but we also need to think about taphonomy if we're going to be asking those sorts of questions as well. So how are they preserved? And with that, I'll say thank you very much. And these are the institutions and funding bodies that have helped with all of these different projects. So thank you very much.